Okay, welcome back everyone to our second speaker of the afternoon session, Yalda Mohsenzadeh. Yalda is an assistant professor at the Department of Computer Science and a core member of the Brain and Mind Institute at Western University in London, Ontario. She's a faculty affiliate also of the Vector Institute for Artificial Intelligence in Toronto. She received her PhD in statistical machine learning from Emir University of Technology in Tehran. She then went on to do her postdoctoral studies at the Center for Vision Research at York University, and then also in the Computer Science and Artificial Lab at the McGovern Institute for Brain Research at MIT. She joined Western after her postdoc. I'm reading a list here. So she joined Western after her postdoc, where her group now conducts interdisciplinary research that spans artificial intelligence, computer vision, and cognitive computational neuroscience. Her focus is on investigating human perception and memory using a combination of neuroimaging in humans, detailed behavior, computational modeling, and machine learning. And we are very fortunate to hear what she has to share with us today. So I'll let Yalda go ahead and set up her screen sharing.
Okay, thank you so much for your kind introduction. Let me share my screen. Okay, can you see my screen, Belle? Okay, I assume everyone sees my screen. So thank you very much from the organizers for inviting me today. I'm delighted to be here and talk about my recent research and on the role of brain recurrent processes in visual recognition. In my lab, we work on basically on interdisciplinary research at the intersection of artificial intelligence and cognitive neuroscience. So we basically study computational principles in the brain, and we use that to inform AI, and vice versa. We use AI and machine learning techniques to understand the human brain. And today, this is like one particular line of research in my lab that I'm going to talk about. So let me start by asking a question, and I will give you a few seconds to think about it. How many vases do you see in this picture? Okay, so let's count them together. 1, 2, 3, 4, and also there is one hidden here, 5. So you see how amazing our visual system is in recognizing objects, even if they are highly occluded and partially visible. But we not only recognize all the objects in this scene, we can also tell a story about it. We can say this person trying to take a picture of this vase bumped to the other ones, and it's about to fall. So our brain automatically predicts what's going to happen in this picture. And this is still a basically difficult task for current AI and computer vision systems. For example, when I feed this image to one of these AI computer vision system, this is what it predicts: a person sitting in a room, which is not so impressive. So what I want to say is that, yes, currently computer vision and AI systems are working very well and in many tasks, but still there is a gap between how we humans perceive the visual world around us versus how computer vision systems do that. Today, I'm going to mostly focus on visual recognition, specifically object recognition, and mostly what we know about object recognition, which mainly happens along ventral visual stream, is from primate studies in, for example, monkeys. And you heard about it, like, for example, from Jim's talk yesterday. And what we know about this visual pathway is that it's hierarchical. It processes visual information along a hierarchy of regions, starting at the back of the brain in primary visual cortex, or V1, which is responsive only to low-level features in images, such as edges, oriented lines, spatial frequency. and it responds around 40-60 milliseconds after stimulus onset. And then at the end of this hierarchy, we have inferior temporal cortex, which is a region known to be involved in object recognition, invariant object recognition. And for example, it can tell us apart faces versus other objects at around 100-150 milliseconds after stimulus onset. While we have like basically a good understanding of these processes in primates, to me still like this picture in humans is less clear. There are studies in using EEG or MEG that they showed that object categorization in the brain happens around, in human brains happen around 150 milliseconds, for example, but still the timing, the processes, the stages that builds up to this categorization is poorly understood. And what we try to understand in my lab is if we can characterize the locus and timing of these steps in human ventral visual stream. For this, we use brain imaging techniques and computational modeling. For brain imaging, we mostly use two specific neuroimaging techniques: functional MRI, which is basically taking pictures of like blood oxygen level dependencies from the brain with MRI scanner, and it has a very good spatial resolution. It can tell us with millimeter resolution where in the brain neurons are activated. This is an example of such picture showing like some blobs of activity in some visual regions. But when it comes to temporal resolution, this signal, this bold signal, is sluggish, and its temporal resolution is very low. It's around like seconds to get to the peak. On the other hand, we have other brain measurement techniques like magnetoencephalography or electroencephalography that they give us a good picture about the brain in terms of timing. So the data from these systems are in form of time series with very high temporal resolution, 1 millisecond, half a millisecond, or even higher. And they can tell us with this high resolution when things are happening in the brain, but nothing is perfect. Their spatial resolution is low, it's in order of centimeters, and it's not comparable with functional MRI. And today I'm going to also talk about a method that can connect these data from different modalities to give us a high spatiotemporal resolution picture of what's happening in the human brain. So today the focus of my talk will be on a series of studies that we have done to study neural dynamics of object perception under challenging visual conditions in the brain. And what I mean by challenging visual conditions is like I show you a demo here, and I would like you to, so you will see a movie that it shows images presented very fast. I ask you to look at these images and see if you can recognize a face image in these sequences. Okay, are you ready? Let me start. Okay, there was a face in this sequence. You can think about, we can try it again. This is another sequence. Okay, there was also a face here, another one. There was no face here. So what I showed you in this demo, this was one of the tasks that we asked our participants to do, was a very fast presentation of 11 images in sequence, and the middle image in the sequence could be a face or not. And participants' task was to detect if they see a face in the sequence or not. This paradigm is called Rapid Serial Visual Presentation of Images, or RSVP. It's been used frequently in psychophysical studies to investigate what level of understanding can be achieved with a very short presentation of images. And as you saw in the demo, and hopefully you also could detect the faces, our participants did very well in behaviorally in this task, and they could detect the target image with significant performance in very high presentation time of 17 milliseconds per picture or 34 milliseconds per picture. The images that was used in these sequences were either selected from this set of face images or these object images as the target, and the masks were drawn randomly from this set of, again, object images. And the question that we are looking to answer is like, how does our brain solve object recognition under this challenging visual condition? So while participants were doing this task, we collected MEG data and we analyzed the data with multivariate pattern analysis. So to explain this briefly, we collected MEG data while participants were doing this task. Then we extracted the sensor MEG data at each time point corresponding to each target image. We showed these target images multiple times to participants, so we have multiple examples of these vector patterns at each time point, and we use a machine learning classifier, SVM classifier here, to distinguish between these target images based on the MEG data at each time point. And we use the performance of this classifier as the distance measure between the conditions. Here are the target images that we have, and we formed a representational dissimilarity matrix at that time point. So you heard about like representational similarity analysis yesterday from Merrick and Moore, so I'm not going to go into the detail, just to explain briefly. So these are matrices, each row indexed with one of the conditions, and each column is with the same conditions, and the element in the matrix shows the distance between those conditions based on the data which could be, for example, here MEG. So we can create one of these representational dissimilarity matrices at each time point using the MEG data. These matrices are very informative to show you like what they capture. I made movies of from the RDMs of MEG data here that you see the RDM on the left side, and multidimensional scaling visualization in 2 dimensions of this RDM on the right side. Faces, so the 2 categories that we have for targets are faces versus objects. Faces are shown with red and objects with blue circles. And so let me play this. So these are like RDMs over time, millisecond by millisecond. As you see in the beginning, it has a random pattern, no specific pattern, but then over time, faces clusters together and separated from objects. I find always this like amazing how information in the MEG data evolves over time to do this categorization. We had 2 RSVP conditions, 34 milliseconds per picture and 17 milliseconds per picture, which is even higher rate of presentation. So I show that here too. So again, a random pattern in the beginning, then over time, faces start to cluster, and then again separated from objects. So you see, like in both conditions, we get these clustering of object versus faces. So having this, so we had also a control condition when images were presented for 500 milliseconds, and there was enough time for participants to see those images. And we had like RSVP condition of 34 milliseconds per picture and 17 milliseconds per picture, as I explained. And we can think about like different ways that temporal dynamics of this categorization can happen over time. The first thing that we would expect is that as we make the visual condition more challenging, so the rate of the presentation is lower, the decoding accuracy will become also lower. So for shorter presentation time, you will have lower decoding accuracy in classification performance. But the peak latency of these decoding curves can have different pattern. For example, they can happen all at the same time, or we can have like peak happening earlier in time for shorter presentation time. So basically, if the presentation of the stimulus is interrupted with the mask, one possibility is that also the peak of this decoding also happens earlier in time, and this can be consistent with like a feed-forward path of processing. And the other possibility is that while we have a shorter presentation time, the peak latency of the decoding happens later in time, and then this will not be only explained with a feed-forward path of processing, and it may indicate some recurrent feedback processing being involved in the process. Let's see what we see in our data. This is the decoding time series over time for the three conditions that we had. First, as we expected, the decoding accuracy is lower for shorter presentation time. But when we look at the peak latency, where basically, which is the time that we have the maximum categorization of the two categories, we see that the peak latency happens later in time for shorter presentation time. This is consistent with the third hypothesis that I explained in the previous slide, meaning that this reduced visibility with higher stimulus presentation rates increases recurrent processing demands in the visual cortex. And it basically shows like involvement of recurrent processes in these processes. We further were interested to better characterize this, like in terms of like spatiotemporal dynamics. So we use the method, voxel-wise fMRI MEG fusion method, and to explain this very briefly, and I'm happy to answer questions about it later offline or like in the questions. In a similar way that I explained, we can create these RDMs from MEG data at each time point, having these RDMs from MEG over time. We can also collect fMRI data while participants view this set of images, and then run a searchlight analysis in the fMRI data and create these RDMs at each voxel in the brain. So we will have like basically these representations over time and over a space that then we can try to compare them over space and time and find like where and when they match, with simply computing like a Spearman correlation between them and finding the significant correlations. This will give us a high spatiotemporal picture of what's happening in the brain, and in form of movies that I'm going to show you here. So here on top, you see the movie that I created with the for the control condition, and at the bottom you see the movie for the RSVP condition. And when I play it, you will see these significant correlations over time and over voxels in the brain. So what you see, like here, like these significant correlations starts at the back of the brain. It moves forward along the ventral stream for both condition. But in the RSVP condition, it disappears in early visual cortex, and then it shows again the return back of this flow of information to early visual cortex, and the timing is also consistent with what we see in terms of peak latencies of categorization that I showed with MEG data. So this is basically the really the first one work that shows these dynamics of feed-forward feedback processing at this high spatiotemporal resolution in the human brain. Let me play this again in case you missed it. So you'll see like it starts again at the back of the brain, moves forward along the ventral stream, and it disappears and then return back. Okay, so to summarize, so far I talked about like how we can map the spatiotemporal dynamics of feed-forward and feedback processing in the ventral stream using the RSVP paradigm and fMRI-MEG fusion method. These findings suggest recurrent computations help categorization in challenging visual conditions, and they can inspire new computational models with recurrent architectures. In the beginning of the talk, I also talked about like how good we are in recognizing objects, even if they are highly occluded. The next question would be, how does our brain solve object recognition under occlusion? So to answer this question, we designed an experiment. We put together a stimulus set of, again, images in 4 different categories and 3 levels of occlusion: no occlusion, 60% occlusion, 80% occlusion. So occlusion is modeled with these black circles randomly on the images. And then again, we collected MEG data while participants watched these images briefly, and they tried to recognize, like, basically if it's an inanimate in the image. This is, I must say that this is a joint work with Saeed Mahdi Khalek and other collaborators. So we collected MEG data, and we again did the same MEG decoding analysis that I explained earlier. And for these 3 different levels of occlusion that we have: no occlusion, 60% or 80% occlusion. And here is the result that we get. So it nicely replicates what we saw also in the RSVP conditions. So first, the decoding accuracy drops for higher levels of occlusion, but also the peak latency happens later in time as we increase the level of occlusion. Again, this indicates that some sort of recurrent processing being involved in helping recognizing objects under occlusion. Then we ask if we know that, for example, that ConvNet, convolutional neural networks, are loosely inspired by the hierarchy of processing in ventral visual stream, and they are feed-forward models. And we ask if a feed-forward model can explain object recognition under occlusion. So we first test this model behaviorally. Here you see the performance of humans in recognizing objects in these 3 levels of occlusion. We use AlexNet trained on ImageNet and test it on these images that we have in our stimulus set. And here's the performance of this model. So while it performs well on occluded images, its performance drops significantly for various as we increase the levels of occlusion. So a feed-forward model is not close to what humans do in terms of object recognition under occlusion. What about brain data? So if we, as I explained, we showed these images to participants, collected MEG data, and we can again create these RDM matrices over time from MEG data. In a similar way, we can fit these images to the deep neural network model and create this RDM from the neural network model. And then comparing this, we will get a correlation time series over time that relates the representation in the model with the brain data that we have. This comparison for feed-forward model is shown here. As you see, while the model shows significant correlations with the brain data when images are not occluded, its correlation is almost zero for the occluded conditions. So the next question that we asked was if a model with recurrent connections can explain the data. So feed-forward model was not good in explaining the data. What about a model with local recurrent connection? Can this model explain the behavioral or neural data? So we got this model from Liao Puju, and we tested this first behaviorally. So this was the performance of the feed-forward model. Now, if we test the recurrent model, we see that its performance is improved for object recognition under occlusion, and it's closer to human performance. In terms of neural data, comparison with neural data, and this was the performance comparison of feed-forward model for when we compared the recurrent model, which is shown with warm colors here. So you see, like, yes, for the unoccluded condition, it shows again significant correlation, but also for the occluded conditions, now we have significant correlation with the MEG data. Okay, so a model with local recurrent connections now can explain both behavioral and neural data under occlusion. And to summarize this second study, we showed that object recognition under occlusion requires recurrent processing, and a feed-forward model cannot explain behavioral or neural data in object recognition under occlusion, but a recurrent CNN model explains both behavioral and neural data under occlusion. In the next study, we ask what is the functional role of these feedback processes in the ventral visual stream? Can we learn more what they do in terms of helping object recognition? So to answer this question, we developed a model, an adversarial autoencoder, and we hypothesized that this model, the generative part of this model, which is an autoencoder shown here, the encoder subnetwork models what's basically represented in feed-forward pathway in the ventral stream, and the decoder subnetwork functions similarly to feedback processes in this pathway, like basically helping to reconstruct more details from images. So we trained this model with super category dataset that we put together, and it was a dataset of including balance, like images of faces, objects, scenes, and animals. And then we also had this data set that's basically includes MEG and fMRI data, while participant viewed images of similar categories. There were like basically the similar set of this data set, which is also, I must say, publicly available for people who would like to use it, of faces, animates, objects, and scenes. So it includes 156 stimulus images, and we collected MEG and fMRI data while participant viewed these images. So we use that data set as a point of comparison with the model that we developed. First, here I show the performance of the model in terms of reconstruction on the test set, our test set, which is basically a stimulus set that's been used for the neuroimaging data. And as you see, it performs fairly well in reconstruction. And we again use representational similarity analysis to make the connection between the brain data, fMRI, in basically making these RDMs from fMRI data at each region of interest, or creating RDMs from layers of the model, or creating these RDMs from MEG data over time, and then comparing them to find the spatiotemporal similarity of the model with the brain data. Let's first look at the spatial correspondence between the brain and the model layers. So we chose two regions of interest, EVC, early visual cortex, and inferior temporal cortex. Here you see the RDM from EVC and its multidimensional scaling visualization, which shows a random pattern. And from IT, here it's RDM, and it's multidimensional scaling visualization again, which shows, again, nice clustering of faces and animes versus object and scenes. And we created RDMs from the activation patterns from each layer of the encoder and the decoder subnetworks, and then did compare them with the brain data in EVC and IT by computing their correlations. So first thing that we observed was that we had higher correlation with EVC in early layers of encoder compared to later layers, and with later layers of the decoder compared to its earlier layers. And that makes sense because early layer in the encoder stack more low-level features, and then in the decoder, the later layers are basically trying to reconstruct the image, so they have more low-level feature information or details about the image. Interestingly, we also see that the correlation of the decoder subnetwork is generally higher than the encoder layers, and that suggests that this reconstruction process that's happening in the decoder is more similar to reconstructing details in the image. That's something similar to what's happening in EVC. In IT, we see that the representation similarity increases along the layer of the encoder and decreases along the layers of the decoder. Again, that's expected because we know from also previous works that these deep networks map the low-level features in the image to high-level categorical concepts along their layers. And here we again see that encoder has more similarity with IT in comparison with the decoder, which is again interesting. So it shows that like the function that encoder is trying to do is like mapping the data to more high level categorical information compared to the decoder or reconstructor. What about temporal correspondence? So we also compared MEG data with the representations along the layers of this model. We see significant correlations both for encoder layers and the decoder layers. But when we look into it in more detail, specifically we look at the peak latency of these curves, we find that the peak latency increases along the layers of the encoder, and it continues to progressively increase along the layers of the decoder. So it shows like this temporal progression along the layers of encoder and the decoder. But more interestingly, we identify two peaks in encoder, which the second peak was, it's also shown here, was basically negatively correlated its progression with the layers of the decoder. So basically this second peak along the encoder layer is happening later in time for earlier layers, which to me is very interesting, and it suggests that this later representation in MEG data is reversing back along the layers of the encoder, which could suggest some that functional reconstruction process is happening here. Of course, we also tested some alternative models. Two important factors in models, in deep neural network models, are architecture and training procedure. So we tested a vanilla autoencoder and for comparison with EVC and IT, and we don't see this pattern that we saw in the adversarial autoencoder. We also tested an untrained model, and again, we don't see that pattern. We don't see even significant correlations. That's the same for MEG data comparison with these two alternative models. So they don't show the specific pattern that we see with the adversarial autoencoder model that we develop and train. Finally, to summarize, using basically by developing this generative model, we are able to dissect the dynamics of processes in ventral visual pathway into two temporally distinct stages, feed-forward and feedback processes, and which suggests the functional role that these feedback processes having in the ventral stream could potentially be reconstructing more detailed information about the visual stimuli that we see. And by that, I would like to conclude my talk with acknowledging my collaborators, co-authors, as well as funding agencies. So thank you.
Thank you so much, Yalda. That was such a cool talk. I really loved that parallel between the effects of occlusion that were happening like in time and space, and then like being able to explain them by adding like recurrence to that feed forward, even that first feed forward visual stream. That was such cool work.
Thank you.
I think you have quite a few questions and quite a bit less time with which to answer them, unfortunately. But it looks like the top question is, isn't learned top-down feedback with recurrence enough to generate these results? I'm not sure which results specifically are being referred to, but there's a lot of additional questions under here. Does that make sense to you, that question?
So is it, I'm not sure if I can get it correctly. So is it about like the difference between top-down feedback versus recurrent, local recurrent feedback, if I understand correctly? So what I can add is that like in terms of modeling, what we tested was the model with the local recurrent connections, and something that's missing to me is like the long range feedback connections in the model, which we also aim to investigate further. And if you remember from the results that I showed you, like under occlusion, we got some boost in like explaining the brain data with the model with local recurrent connection, but still there was a gap to get to the point to explain the model, like the performance when the data is not occluded. And to me, that gap can be filled with like those type of feedback connections. Okay.
Cool, thank you. So it looks like the next question is one of mine. So I'm wondering about the clustering of faces versus objects in the rapid serial visual presentation task. Do you think that that's an inevitable preference for categorizing faces versus other objects, or is that a function of the task?
Okay, that's a very good question. So to me, the timing of what's happening, like it was pretty early, like before 200 milliseconds. So it's more related to like automatic categorization that happens in the brain quickly in ventral stream, rather than the obvious connection with behavior, because I must say that it was a delayed task too. So like it's if it was like really a detection basically related phenomena, it would be a bit later. And this is something that we see it in many sets of data, even like if the participants are not doing any task, they just passively viewing images, we see around the same time the categorization happening. So it's more like to me, the automatic process is happening in ventral stream.
Awesome, thank you. So it looks like we probably have time for one more. So this is a question from Christopher, who says, big fan of the fusion analysis, exclamation point. One problem with the fMRI time uncertainty is that the bold activity in EVC might already be composed of initial processing as well as the recurrent later influences from higher sources. Have you looked at MEG signal maybe in source space to ask if you can replicate this feedback stream with Granger causality, for example?
Mm-hmm. Yeah, very good question, and we always do that. Yes, that's one of the limitations. And when we are using the fusion method, we always look at the source localized MEG data, and to see like the consistency. And in this case, we see it. And in the eLife paper that we published on this work, we also did fusion of source localized MEG with fMRI data to make it even like more stronger in terms of like capturing the representation from a specific region rather than like combined representations.
Awesome. Thank you so much for an excellent talk today, Yalda. It was really cool. Learned all kinds of new things.
Thank you. Thank you for having me. It's been a pleasure.
Yeah, our pleasure. So up next, we're going to have Zach Pitkow coming to talk. So I would encourage you to come back in probably 2 or 3 minutes when hopefully we'll have Zach